Hello physiologists. We've discussed the digestion in the small intestine in another video, and in this one I'd like to talk about absorption in the small intestine. So the goal of digestion is to take these large food molecules and break them all the way down into their smallest monomers so that they're small enough to move through an absorptive cell in the small intestine and then into either a blood or a lymphatic capillary. Carbohydrates need to be broken down all the way into their monosaccharide basic forms. Proteins need to be broken down all the way into single amino acids, ideally, although you can get dipeptides or tripeptides through. Triglycerides need to be broken down into their individual fatty acids, glycerol, and monoglycerides. So those are the monomer forms of each of these major nutrient groups. For monosaccharides, so these are your smallest, your building blocks of your carbohydrates. All dietary carbohydrates that are digested are absorbed. Your body is very efficient and very good at getting every single possible energy molecule that you could use for fuel, for ATP, later. The only things that your body can't digest and absorb are indigestible cellulose and then some of the fibers in plant matter. And so these are going to be left in the feces. Monosaccharides are going to be absorbed through facilitated diffusion or active transport moving into the blood. When we look at proteins, amino acids, dipeptides, and tripeptides are your smallest forms that we're looking at. Mostly, they're going to be broken down all the way into single amino acids. And then they are moved through active transport into blood. Remember that active transport means it requires energy of some type. Half of all of the amino acids that you absorb actually come from the proteins in your digestive juice and the dead mucosal cells. Digestion is a little bit rough on your stomach. You end up shedding a little bit of the lining every time you digest a meal, and you also have tons of enzymes that are being secreted, which are proteins. And so as you move toward the end of digestion and you have these leftover enzymes that aren't being used anymore, you can break them apart and you can absorb those as well and reuse them. Let's take a closer look at carbohydrate absorption. So what we have here is one of the absorptive cells of the small intestine. So we're zoomed in very far, and we're just looking at one cell in the small intestine. So if we take a closer look at this cell, we can see that it does not look the same on one side as the other. So this bottom part of the cell, this is the basolateral membrane, and then you can see there's a blood capillary right here. So here's the blood supply, and these nutrients are heading into the blood supply past the basolateral side of the cell. And then over here we have the apical or luminal surface. And you'll notice that all the microvilli that are present in the small intestine, we see those on the apical or the luminal side of this cell. So this area over here in this kind of pinkish beige color is going to be the lumen of the small intestine. This is where all of your chyme is and also where all of your enzymes are mixing with your chyme. And so ultimately what we want is after our nutrients are broken down to their smallest monomers, we want them to be moved from this lumen out here in this pink beige area. They need to go all the way through the cell and then through the other side of the plasma membrane and ultimately into this blood capillary right here. So we're going to take a look at some more specifics of that, but it helps to make sure that you can orient yourself. Basolateral side, down here near the blood, and then you have the apical side over here with all of the microvilli. This will be on the side by the lumen. Okay, so the first thing we need to look at is this sodium-potassium pump right here. So hopefully you're familiar with the sodium-potassium pump. It moves sodium through one direction and potassium to the other direction. And this is a great example of primary active transport, which means it requires energy, and in particular, ATP is the energy that it needs. So sodium is constantly being moved out of the cell, and potassium is constantly being moved into the cell. And I want to focus especially on this sodium. So whatever sodium happens to be on the inside of the cell is constantly being moved out of the cell because of this sodium-potassium pump, or sodium-potassium ATPase. So if you're moving sodium constantly out of the cell, that means that you are going to have a low concentration of sodium inside the cell. So you have established a concentration gradient because of the sodium-potassium pump that's here. And that's going to be really important because when we move up here and look at the apical side, you have a sodium-glucose symport transporter. Now this is another example of active transport, but it's secondary active transport. And the reason for that is it requires energy, hence the active transport, but it uses something different than ATP. And in this case, we're using sodium, a sodium gradient. So if you look at this picture, as sodium moves into the cell, it takes other things along with it. It can take glucose or it can take galactose. And so glucose and galactose are making use of this sodium gradient that was originally established by our sodium-potassium pump. As sodium moves into the cell, so also does glucose or galactose. And this pump must have that sodium gradient established in order for it to function. If you get rid of this sodium gradient, if you have the same concentration of sodium on the luminal side as you have on the inside of the absorptive cell, then this symporter cannot work. And symporter means you have movement in the same direction of this molecule. So sodium is moving in one direction, and then glucose or galactose is moving in the same direction. So that's why it's a symporter. But if that sodium gradient gets messed up, then you're not going to be able to transport your glucose or galactose. So this sodium gradient that's established by the sodium-potassium pump down here on the basolateral side is very important. So as you move sodium in, you also move glucose or galactose. And then you also have facilitated diffusion that's possible for fructose. Now diffusion does not require energy, but facilitated diffusion means even though no energy is required, you still need a transporter. Fructose does not have the ability to diffuse across the membrane by itself. It has to use a carrier. And so you see this transporter right here, but because it's diffusion, there's no energy required. So fructose just diffuses. It's a little bit different than glucose and galactose. Diffuse through this transporter. And then all of your sugars, glucose, galactose, fructose, they travel through the absorptive cell all the way over to the other side to this GLUT transporter. And it is able to move all of these sugars, all of these monosaccharides, to the other side of the membrane. And this is another example of facilitated diffusion. So no energy is necessary, but a transport protein is required. So all of these sugars, as they're passing across the basolateral membrane, are moved through this GLUT2 transporter. And then from there, these sugars, these monosaccharides, can then just diffuse into the nearby blood capillary. Remember, in each villus of the small intestine, you have a blood capillary and a lymphatic capillary right there. And so it's easy for nutrients to make their way into the blood or the lymphatic system from here. And if we're talking about carbohydrates, that will specifically be into a blood capillary. Protein digestion and absorption is a little bit more straightforward. So if we look at this general process, here we are in the mouth. You consume this protein, maybe some meat, maybe some beans or cheese. In the mouth, you don't have any chemical digestion occurring, just the mechanical digestion where you mash it up. When you get to the stomach, now you have hydrochloric acid, which can denature this protein, start to unfold it, and you also have pepsin that can start to cleave some of these peptide bonds. And so you can see in the stomach, you end up with shorter proteins, shorter polypeptides right here. When you get to the small intestine, you have even more proteases. So you have trypsin that is available in the lumen, you have chymotrypsin, and you can see it with these arrows, you're continuing to cut these polypeptides into smaller and smaller pieces. And so now we're down to 3 or 4 amino acids per polypeptide. You also have some carboxypeptidase that can start removing 1 amino acid at a time. So thanks to the enzymes from the pancreas, you start to further break down and shorten those polypeptide chains. When we have digested our protein down far enough where we just have a couple of amino acids stuck together, that's where our brush border enzymes come in. And our brush border enzymes, such as aminopeptidase or dipeptidase, these are able to cleave off those individual amino acids. And once we have those individual amino acids, those are able to move through the absorptive cells one by one and make their way into the bloodstream. So protein absorption happens with individual amino acids after they have fully gone through the digestive process. Lipids need a little bit of extra help because they are in a watery environment in the chyme, and then the blood is also a watery environment, and lipids don't dissolve very well in water. They're able to get past plasma membranes pretty easily because the plasma membranes, their nickname is a lipid bilayer. They're made of primarily phospholipids. So lipids can cross plasma membranes relatively easily, but once you get into any kind of watery environment, they have some trouble. So your dietary lipids will be absorbed by simple diffusion for the most part. Their short-chain fatty acids will go into the blood for transport, but the long-chain fatty acids and the monoglycerides, so that is one fatty acid, mono, combined with glycerol. So if you remember your structures, you have triglycerides, you have 3 fatty acids, tri, bound to glycerol, so triglycerides, 3 fatty acids and glycerol. So you can take 2 of those off, and then you'd be left with a monoglyceride, for example. But these fatty acid chains and monoglycerides are pretty big, and they're also hydrophobic, so they don't dissolve in water very easily. And so what we're going to use are our bile salts. So those secretions from the gallbladder and the liver are going to come in handy right now. So bile salts make my cells that act like ferries for not just the fatty acids and the monoglycerides, but also for fat-soluble vitamins. So vitamin A, vitamin D, vitamin E, and vitamin K. And the micelles can act as ferries to kind of help transport those lipids to the absorptive cells. And then once those fats, those monoglycerides and fatty acids, once they get to the absorptive cell on the inside, they reform into triglycerides. All of your energy molecules have a fantastic ability to be broken apart and then reform and be transformed into other molecules. So once you're in the absorptive cell, they'll turn back into triglycerides, and then they're going to get coated with a protein, and then they'll be called chylomicrons. Then they're able to leave the absorptive cell by exocytosis, and then from there they're going to enter a lacteal. These fats are too big to be able to get into one of the blood capillaries, so they're going to go into a lymphatic capillary instead. And the lymphatic capillaries that are present in the digestive system that are used for lipid absorption are called lacteals. Now all of the lymph from the lacteals, including these fat-soluble vitamins and lipids, these ultimately will enter the bloodstream once you get to the subclavian vein. So eventually you will have these fats that enter the bloodstream. They just kind of need to take the scenic route around. The chylomicron coat will help them as they're being transported in the blood because the chylomicron coat is protein and that is much more water soluble. So here is an image of what that lipid absorption looks like. So one of the things that bile can help with is emulsification, which is where you break apart a large fat globule, as you can see in the top over here, and you break it apart into smaller fat globules. And here's your bile acid and lecithin. So here you have smaller fat globules right here. This increases the surface area. So now, with these smaller fat globules, pancreatic lipase can act, has a lot more surface area to act on more bonds. And so they can help this triglyceride. So here's glycerol right here. And then you have 1, 2, 3 fatty acid chains. And so pancreatic lipase will turn this big triglyceride into this. Here's some fatty acids here and here. And then here's your monoglyceride, glycerol and a fatty acid chain. So pancreatic lipase will actually cleave the bonds, but it's made more effective thanks to bile increasing the surface area and breaking up those large fat droplets. Now you can use micelles to transport your lipids. So here's your monoglycerides. You use a little bit of cholesterol, fat-soluble vitamins, fatty acids. So here are your lipids, and they are going to form the core of these, and then they're going to be covered in bile acids. And so these micelles, I think of them as ferries. They'll be able to transport these lipids, as you see down here. They transport the lipids to the absorptive cell. And notice that the bile salts are not going into the cell. The bile salts will remain in the small intestine and eventually go to the large intestine and be excreted. And remember, this originates from bilirubin in the liver. And so this is how you get rid of bilirubin. You excrete it in this way, but they have a really good use until then. So micelles act as ferries, moving these lipids to the absorptive cells, and then the bile salts can go help form more micelles and bring more lipids to the absorptive cells. And then you can see in here, here's your fatty acids and your monoglycerides. We just broke them apart, and they're just going to reform. Here's your triglyceride again. Everything has been put back together. We've got Humpty Dumpty over here. And they combine with some phospholipids and some cholesterol, get a protein shell on it, and here's your chylomicron. And so once you've made your chylomicrons, they go through Golgi apparatus. Remember, Golgi is kind of like your post office of the cell. It packages up whatever you need, and it sends it to the right place. And here are your chylomicrons in these vesicles, and they leave through exocytosis. And from here, they can go into a lacteal, which is a lymphatic capillary. And you'll travel in the lymph until you eventually come to the subclavian vein, and then that will join your blood. So lipid absorption requires the use of bile salts, the secretions from your liver and gallbladder. So here is a quick summary of chemical digestion and absorption in the digestive tract. It is not all inclusive, but it does provide a pretty good overview. And it's divided up based on which type of molecule you're looking at. So here's carbohydrates, you have proteins in the middle, and then fats on the right-hand side. And then it's also divided up based on where certain enzymes are secreted. So you have the mouth, stomach, small intestine, and then finally where those energy molecules will end up on the bottom. So carbohydrates begin digestion in the mouth. You have salivary amylase that begins breaking them apart. There's not really any carbohydrate digesting enzymes in the stomach, but once you get to the small intestine, you have pancreatic amylase and then several brush border enzymes that will continue to break down those carbohydrates. Those monosaccharides travel through the absorptive cells and ultimately end up in a blood capillary. When you're looking at proteins, not much digestion can occur in the mouth other than mechanical digestion, the mashing up with your teeth and tongue. In the stomach, you have pepsin that will help to start break down the proteins. Not pictured is hydrochloric acid as well, which denatures the protein. When you get to the small intestine, you've got trypsin, chymotrypsin, carboxypeptidase, thanks to the pancreas. And then you also have a couple of brush border enzymes that will ultimately digest those peptides down into individual amino acids. They are transported through the absorptive cells, and then they also will end up in that blood capillary. For fats, you don't have any digestion chemical that happens in the mouth, but once you get to the stomach, when you swallowed that lingual lipase, it begins to work in the stomach. You also have gastric lipase secretion. You start the breakdown of your fats. When you get to the small intestine, you've got pancreatic lipase that further breaks down those fats, and then you also have bile and bile salts that are secreted from the liver and the gallbladder that help you. So those micelles will help move your broken down lipids, your monoglycerides, and your short-chain fatty acids into the absorptive cells, where they will then recombine and create triglycerides. Remember, your bile salts will remain in the small intestine lumen and eventually be excreted. But you do have your lipids or lipid-soluble vitamins in here, cholesterol, phospholipids, any of those things that are of use to you and your body. They will be packaged up into chylomicrons, get that protein shell on them, and then they leave through exocytosis to go into a lacteal, which is a lymphatic capillary. And eventually, these will make it back to your blood, but initially they'll go into that lacteal first.